Hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled Biomarker Discovery, Integrating Proteomics and Genomics for Comprehensive Biological Insights. My name is Corey Stanton and I'll be your X-Talks host for this event. At this point, I'd like to thank Discovery Life Sciences who developed the content for this presentation. Discovery Life Sciences is a global leader in biospecimens and specialty lab services, enabling the discovery and development of groundbreaking therapeutics and diagnostics. With a robust portfolio of human biospecimens and cellular starting material, preclinical ADME tox solutions, and cutting-edge specialty lab services, they provide their partners with the tools, data, and expertise they need to succeed. From genomics and proteomics to cell biology and molecular pathology, their comprehensive service offerings support every phase of the scientific journey, from research through clinical trials. And now let's take a moment to meet our featured speaker for today's presentation. Dr. Jessica Moore is an expert in mass spectrometry and proteogenomics, specializing in integrating high-plex proteomics data with LC-MS datasets to advance multi-omics research. She develops innovative sample preparation methodologies and non-invasive biomarker discovery approaches, collaborating across disciplines to deepen understanding of disease progression and molecular mechanisms. By bridging proteomics with genomics and transcriptomics, she translates omics data into actionable insights, driving precision medicine and industrial applications while fostering collaborative partnerships. And now, without any further ado, I'd like to hand the mic over to Dr. Moore to kick off the presentation. Jessica, I'm going to send you screen control now, and the floor is yours.
All right. Well, thank you all for joining me today and the opportunity to speak to you about this exciting topic. And how we are using a multi-omics approaches for biomarker discovery here at Discovery Life Sciences. So in today's presentation, we have a couple of key learning points to talk to you about today. And then in this, we're going to first emphasize the importance of study design when we build omics cohorts. Next, we're going to focus on minimally invasive biomarker discovery and its power in clinical settings for monitoring and longitudinal studies. And finally, how we integrate omics data to help us tell data stories that deliver more actionable results. Now, this webinar is focused on biomarker discovery. So to begin, let's talk about some common definitions. Biomarkers are measurable signals that are indicative of a biological state or disease. And any class of biomarkers, biomolecules like metabolites, lipids, proteins, RNA, DNA, they can be used as a biomarker. And they can be used for diagnostic, prognostic, monitoring, risk assessment, pharmacodynamics, safety-related monitoring, any of these. And we can use multi-omics approaches in order to do some of these studies. And that can include looking at multiple omics together. All right. And so that's one of the things that we're going to look at in the talk today. We use multi-omics analysis to deepen our understanding of these systems, and it serves to advance our understanding of biomarkers and how we use them to diagnose and prognose disease. And this story really starts with the central dogma. The central dogma drives our understanding, and this figure was taken from Crick's 1970s publication in Nature, and it remains an excellent figure even 50 years later. It shows how dynamic the processes are for translation of proteins, and the understanding of this relationship is the reason that we study biological systems with multi-omics. Our end goal is to deepen our understanding and to help our findings become more translatable to actionable results. This looks a little like this. Actionable results are our end goal. Typically, these studies, they begin with a cohort, human, mouse, other, and we collect data and we begin to analyze it either with supporting independent results or true integration. We'll talk about that a lot. And it results in the generation of a signature that we can validate and we can use for diagnostic and prognostic decisions, pharmaceutical efficacy decisions, tox results. It all begins with a cohort. And this slide may be a little iterative, but there are many times when having more data is helpful to our biological hypothesis. That's because biology is complicated, it's dynamic, it's often interlinked, and there are compensatory mechanisms that complicate our hypotheses. But the more information that we do have, the deeper our understanding of the systems at that snapshot in time will become, in the snapshot in time in which that sample was taken, and therefore the more actionable results that we can deliver to our colleagues who are either scientists or physicians. Omics study design. It all begins with a cohort, and study design is paramount for multi-omics experiments. Garbage in, garbage out. I'm sure you hear that a lot, but in this first section, we're going to take a step back and really focus on study design and our considerations. A successful omics experiment begins in design and enrollment. Any time that you can spend here in this stage is time well spent. And the work begins prior to enrolling your patient populations. Consider inclusion criteria. What patients are you targeting? What phenotypes? What age? Are there gender considerations? We're going to talk a lot more about associated metadata on the next slide, but enrolling the correct cohort is really mission critical. Additionally, how can we make sure to minimize any pre-analytical variables? If you're collecting blood, is there an SOP? How long can it sit at room temperature before it's centrifuged to become plasma? Will it be collected at various sites? Are they going to be in the same tubes? Can we minimize pre-analytical variables? It's going to remove potential causes of variance. And anytime we can decrease variance, we allow those biological conclusions to emerge from the background noise. Finally, a big topic of discussion is depth versus breadth. How many samples are we studying? You can really build a solid biomarker discovery cohort in two ways. We can make a very large data set by querying fewer biomarkers over a lot of patients, or we can take a smaller sample size and mine it quite deeply, creating a powered study. This is really a consideration for diseases with low incidence rate, where it's extremely difficult to enroll a large cohort of patients because your phenotype is rare. And so we can come up with solutions to help those folks too. Finally, what is our matrix of intended use? What samples are we enrolling? Will it be something like a biopsy that's more invasive? Will it be something less invasive, like a blood product? Urine, a skin tape, a swab, something that's very minimally invasive. And we're going to return to this topic in greater detail in the second section of this talk, where we're going to discuss non-invasive sampling. But when we're talking about cohorts, we really have to talk about metadata. Metadata is incredibly important to consider when building a cohort, and thinking about metadata really starts with the field of epidemiology. What do we know about this disease? Who does this affect? And when we consider the disease and progression and burden, where in this timeline do we want to interrogate? Consider biomarker discovery for early detection. If we want to find a biomarker present for early detection, we need to have a sample to interrogate of people who will go on to develop the disease. Let's think about a neurodegeneration cohort. Maybe the best people to test for early detection are pre-symptomatic people, but we can't really sort them into their classes of disease as yes, no, until they progress to develop the disease. So in that case, a well-designed cohorts and those types of hypotheses, they might take decades to collect. And it could be even longer if you need to power the study. And the more time that it takes to enroll the cohort, the more those pre-analytical considerations from our last slide come into play. What happened to that sample over the last 20 years? Were there power outages? Were there freeze thaws? Another thing that you might consider is this. What are you hoping to compare? Many times we aren't necessarily considering a diseased population versus healthy. Many times we're considering a disease and a clinical mimic. Is it appropriate in, say, a skin cancer cohort to compare a lesion to normal skin? Or is it appropriate that your control be a mole that is benign? So in addition to comparing the correct biological states or disease conditions, we have to match them for more confounding variables: race, ethnicity, socioeconomic status, age, gender. It's for sure a lot to consider to design a cohort like this, but it's really fundamentally important. And there are two big takeaways from this slide. One, associated metadata is really important because it represents a potential source of additional variability. And two, the study size and groups must be appropriate both to your hypothesis, to power your study, and based on the tests that you want to do. All right, so let's simplify this a little bit to just a cohort of control and disease and expand our potential omics. If we were to consider genomics as the foundation, the blueprint of life, and all the downstream omics as more dynamic, they're a snapshot in time. And because they are dynamic, studies following disease progression or drug efficacy likely need a multitude of samples to represent a single patient's journey through a disease or a treatment. It's a snapshot in time, and we might need a multitude of snapshots in order to capture our biology. Finally, we know that poor experimental design makes our bioinformatics complicated. Garbage in, garbage out. Quality studies really need to consider enrollment criteria, cohort demographics, time points, pre-analytical variability, and analytical variability. Then that's all in addition to their big omics data sets. And I want to leave this section with this idea of really reiterating that good studies are designed with both depth and breadth when possible. In proteomics, particularly within the plasma proteome, we often use this iceberg as an analogy for the plasma proteome to showcase the need for dynamic range. You maybe have seen it before. But if you think about one patient at one snapshot in time as one iceberg, and we need to study the depth of the disease, it also becomes apparent that to truly understand a disease, we need more than just one iceberg. We need to study the entire ice field. All right. I'm going to show you a basic example of an FFPE proteomics data set. This is a single omics, just proteomics, on a small cohort of breast cancer. And this represents a 65 FFPE sample study, so it's very modest, but encompasses a total of 44 unique patients with 19 matched sets. And that includes tumor and adjacent normal breast tissue that we studied using LC-MS, single omic. Even with a single omic studying very deeply, we can leverage a strong sample prep and the speed and sensitivity, in our case, of an Orbitrap Astral mass spectrometer to mine very deeply on a very small cohort. So using next generation mass spectrometry, this study can represent over 10,000 protein groups that we were discovered with an average of 8,626 protein groups from a single FFPE curl, a single 10 micron curl. So the depth of this study is really incredible, especially when we consider the minimal input material. And we can begin to interrogate these data. All right. So this is a principal component analysis that's covered by benign or malignant. And here benign is adjacent normal tissue. It's still a cancer patient. And these regions show clear separation, which is quite impressive when you consider that this is from a single 10 micron curl, single point of time, single surgery, tumor versus adjacent tissues. And there are types of analyses that we can also do with these data. Here, a single omics, we're taking these match sets and comparing the protein from the single patient adjacent normal tissue to tumor, which we're showing here on the left. And we can take the proteins that were identified and perform pathway analysis, which we're showing here on the right. And it's highlighting the pathways that were implicated within the disease. It's really impressive data to deepen our understanding of this disease within a single patient. But let's think about what we might have missed in a study like this. When we consider the landscape, the breadth of depth and of breast cancer, what might we be missing? How could we query this landscape in a way that is more all-encompassing or more specific for, say, a niche type of breast cancer? What variables did we not consider? And those are kind of listed here on the right, things like genetic variants, breast density, all the things that we need to think about. And I'm going to use a statistic here as a transition to the next section, and that is talking about the sheer number of breast biopsies that we perform in the United States every year. It exceeds 1 million, and that's from cms.gov. So that's 1 million humans who endured a biopsy, potential complications, pain, clinical visits, and associated costs, only to, quite luckily, have a majority of them be diagnosed as benign. And that leads us to the second main point of this webinar. That's the minimally invasive biomarker. Because if we can envision our ideal biomarker to help diagnose and prognose disease, we probably don't want it to be surgically derived. We want something that is easy to obtain and it's palatable to the patient, so that they will return for future screenings and follow-ups. And that brings us to this next section, the minimally invasive matrices. And that's really why we're discussing minimally invasive matrices for biomarker discovery within this presentation. So I will allow that a resection is really a gold standard way to study a tumor. We absolutely agree. However, they do come at a cost, both to the patient and to the payer. Now, conversely, blood products are minimally invasive. They're often banked in clinical trials, and they present the opportunity to study a single patient longitudinally. Several other non-invasive matrices also present this opportunity. Matrices like skin tapes for maladies of the skin, urine to watch what's filtered from the blood, and then swabs of the nose, the cheeks, pap smears, lavage type samples. They're all wonderfully underrepresented matrices to study biomarkers. And in this next section, we're going to focus on blood products, but I will absolutely want to point out that these are all exceptional opportunities for biomarker discovery. Now, from a proteomics point of view, plasma proteome is a bit of an analytical challenge, and specifically that's because the top 20 proteins account for 99% of protein content by weight. And that's hard. It's hard to measure. So therefore, we typically use an enrichment approach. That can include direct measurements like mass spectrometry with physical enrichment for low abundant analytes. These products are the Seer Proteograph, Proteomics Enrich, magnet beads, things like that. Or indirect measurements where we use an antibody to an antibody or an activator to target the protein of interest. And that includes things like Olink's proximity extension assays, Alamar, SomaLogic, those types of approaches. Here at Discovery, we use two parallel approaches to interrogate serum or plasma for deep proteomics. Now, that includes Olink Explorer's HT proximity extension assay, which measures a dedicated 5,400 proteins from just 2 microliters of sample, as well as the Seer Proteograph platform tied to an Orbitrap Astral mass spectrometer, which utilizes nanoparticles to enrich for low abundance proteins from complicated biofluids in a completely untargeted and species agnostic way. And we enable deep agnostic data sets by pairing this sample preparation technology with the Orbitrap Astral, who is absolutely excellent at delivering speed and sensitivity for mass spectrometry analyses. And we found that these two technologies are extremely complementary. They allow us to query more of the proteome to maximize mechanistic understanding and to measure very low abundance proteins such as cytokines and chemokines, which are often quite important markers for inflammation and cell signaling. All right. So here in this example, we're going to take a single aliquot of plasma from a patient and split it between Olink Explore HT and the Seer Proteograph with Astral mass spectrometry. So we wanted to build a cohort to show you this. And we started with a moderately sized cohort of plasma samples. This initial cohort contained 410 samples that met quality control for the assays. And this was a pan-cancer cohort representing 3 cancer indications: colorectal, lung, and prostate with age and sex matched normal donors from the Discovery BioStore. So these normal people self-indicated that they had no cancer. And we analyzed these. The first multi-omics example that I'm going to show you in this talk is actually 2 proteomics data sets. When dealing with 2 data sets that are quite large, there are a handful of considerations that we can make. First, we can look at overall identified proteins and see if there are common identified proteins. And this grants us confidence to the measurement if the same proteins are qualitatively present in 2 separate assays. It's a secondary confirmation of the protein. Next, we can look at each data set independently. Here we've performed a simple t-test of normal versus cancer, and we can compare the proteins that are up in cancer versus normal in both assays independently. And if we considered the protein group that might be up in both assays, it's a secondary confirmation. And that can give us confidence that these trends are trustworthy prior to investing the time and energy to validate a biomarker. All right. So integration of omics data is bringing us to our final topic. Previously, I showed you that integrating 2 proteomics data sets is something that's tricky, but when we start to integrate with other omics, we come to this final topping. And it's interesting to note here that data integration of omics, it doesn't necessarily have to be genomics to proteomics, proteomics to RNA-seq. It can actually be a single omic to itself with complementary technologies. And there are a couple of ways to do this. Here is a very basic way to think about this. We can analyze omics 1 independently to conclusions and omics 2 independently to conclusions. And then we can look for common patterns, common pathways, or observations that were independently observed in each omic. Like in the previous example, I do think that this is a powerful approach because it essentially gives you unbiased secondary confirmation of an observation. However, one thing is fundamentally missed, and that's how the central dogma really relies on itself. It's dynamic. It's deeply and truly interconnected. So there are many ways that we could miss things when we don't fundamentally integrate omics. And then integration of these data is the foundation of proteogenomics studies. There are many ways when this is smart to do. For example, let's say you built a custom cell line with CRISPR and you wanted to confirm that your protein was knocked out without any other unintended effects. Proteogenomics. It's an excellent way to do that. It builds you a data story where you can understand a biological system that you built before you take it and you study it. And I'm going to share an example of these first 2 with a tumoroid story that's going to tell a very similar story. All right. Whenever we extract tumor tissue from a patient, it can be stored in a lot of different ways, including fresh frozen biopsies, dissociated tumor cells, FFPE samples, or a tumoroid. What's a tumoroid? All right. Tumoroids can be grown and cultured, and as they grow, they structure themselves into multicellular communities of cancer cells, which is really valuable as a tool, not just to study cancer biology, but also to test pharmaceuticals for efficacy, for dose screening, and other applications. But really, in order for us to know when they're appropriate to use to test some of these things, we have to characterize them just like you would a CRISPR line. But since they're a culturable product, understanding the cell's fundamental biology is quite paramount so that we know what line to pick to fit our research model needs. And this is an opportunity for something like a proteogenomics approach to perform an excellent solution to characterize these lines. And so that's one of the things that we sought to do here. This experiment utilized a handful of proteogenomics analyses on tumoroids. And the experiment was set up to perform whole exome sequencing on a handful of tumoroid lines and then to add LC-MS/MS data to this workflow. We use the Proteograph Analysis Suite, PAS, in order to analyze these data collected in DDA mode. And then we were able to identify over 13,000 peptides, and there were a total of 109 variants. And of note, these tumoroids are in their native state. We didn't stress them in any way. They're just happily growing. So we anticipate that this same type of analysis becomes even more interesting when we put these lines in the presence of like a test article or a stressor. Here are some of the SNP variants from the tumoroid samples and an example of what we can do with these types of data. So first, we looked at the SNPs in terms of what was monitored in the TrueSight Oncology 500 panel, very commonly used. And a majority of these are not actually on the TSO 500. However, if we compared them to this 2024 paper to assess the functional impact of the cells, they're predicted to be impactful in cancer with recovery when that gene's knocked down. Now, obviously, we can hypothesize on the right-hand side about potential effects, but we can also hypothesize by querying the literature that a number of these are known druggable targets. And this example really showcases how we can use proteogenomics data not only to just assess the functional impact of the cells, but also to really guide potential biomarker discovery and drug development. We're going to show you an example of this. So this is a single of the tumoroid lines. It's a colorectal cancer, and we're looking at KRAS, a KRAS mutation from a single colorectal derived tumoroid. And KRAS, of course, is one of the most frequently mutated oncogenes in human cancer. Most of us know about this story already. And in this case, it was a replacement of a glycine with a cysteine, which keeps this protein active, and it drives rapid cell proliferation, telling it to grow. So we can track KRAS, but we can also track several associated protein variants that are downstream of KRAS. So this is an example of a known oncogene implicated in disease progression and then downstream potential oncogenes. And it represents more druggable targets and, more importantly, the potential for combination therapies. So in this characterization, we can look at known therapies, downstream variants, and then potential alternative therapies with combinations. So it's really exciting to be able to see these types of data. All right. So our end goal is always to enhance the translational value of these data. How does the addition of proteomics data enhance genomic data alone? It's probably a question a lot of you are asking. So first, a majority of therapeutic targets are in fact proteins, and proteomics has the ability to validate the impact of genomic mutations as a functional test. Moreover, just like KRAS, there are pathway-driven changes that we can monitor. We know that proteomics data can augment transcriptomics data because there's often discordance between the two measurements, simply because they're a snapshot in time. And combining those omics heightens our understanding at a systems level, and it reveals how genomic mutations drive proteomics and how it can impact the entire system. So let's explore this in a larger cohort. Here's our body again. In this talk, we've established a few key points at this time. Tumors remain the gold standard for studying cancer, and that serum and plasma are really critically minimally invasive matrices for things like early detection and longitudinal monitoring, and they're routinely banked in clinical trials. And so we want to explore the work of these two different matrices together within a cohort. So in the next data set I'm going to show you, we designed a cohort of matched sets where a single patient is contributing both plasma and FFPE blocks. And often the FFPE blocks include a multitude of samples, often representing tumor and adjacent normal tissue. And here we are also extremely cognizant of our tissue use for these precious biopsies. We're using about 250 microliters of plasma and a total of 5 10-micron curls to do this analysis. And if we consider deep omics to study a single patient with multiple omics depth in both plasma, tumor, and adjacent normal FFPE samples, we can make an even bigger data story by powering this study over a multitude of indications and patients, so breadth. All right, this is what we sought to do within this initial study. One, we want to combine Olink Explore HT with the Seer Proteograph with astral mass spectrometry for a comprehensive plasma proteomic profile. And that's going to include low abundance markers like cytokines. Two, we want to perform whole patient profiling utilizing matched sets such as plasma and FFPE. And three, we want to perform and integrate multi-omics analyses to develop potential biomarker signatures. We did this by expanding our first cohort that we showed you. That initial cohort was about 410 plasma samples alone, and we wanted to expand it to samples that included matched plasma, FFPE tumors, and normal adjacent tumors. So that's the secondary cohort here. We enrolled patients within the same 3 indications in both stage 1 and stage 4 of disease, and we began collecting data. These data are still a work in progress. Obviously, it's a big data set, but today I'm going to show you some integration of RNA-seq and a multitude of proteomics data sets. The FFPE samples we sent through RNA-seq using a dual DNA and RNA extraction process, and the RNA was sequenced on the NovaSeq X. When a t-test was performed comparing malignant resections with adjacent normal FFPE, a total of 701 genes were found to be differentially expressed, including 257 up in malignant and 444 down. And here at the bottom is just a listing of the top 10 up and down regulated genes. We can take these data and do a lot of different things with them. Here's a principal component analysis. Showing clear clustering between cancer and adjacent tissues with further differentiation from site of origin. That's always really exciting to see. Within a cancer type, when compared to adjacent normal tissue, there are absolutely differentially expressed genes that were identified. This Venn diagram on the lower right shows that. So this is a t-test of colorectal versus normal, lung versus normal, prostate versus normal, and the things that were upregulated in those comparisons. And then we set it up in a Venn diagram to show you what is shared that is not that is conserved within each cancer type, and those that were unique to specific cancers. So that's a cool data query to be able to do. And here are some key signatures of disease. These that resulted from these data. Here are some of the examples of the upregulated pathways, and they included stress response, trafficking and secretion, proliferation and metastases, immune modulation, extracellular matrix remodeling, and then also metabolic changes. So these are all things that we do expect to see within cancer, and it's really lovely to be able to confirm things like that with these data. Here are 3 examples of transcripts that aligned with our expectations. They're very well studied proteins. Cystatin 1 is known to be overexpressed in cancer and currently is being studied as a prognostic biomarker. So in these data, you can see a box plot for cancer on the left and normal adjacent on the right. And in this case, all 3 cancer samples show an increase in these transcripts, with prostate adjacent normal also showing a bit of an increase. The second example on the right is ASCL2, and that one is also known to be overexpressed specifically in colorectal cancer. And indeed, we detect that in these data with low levels in lung and prostate. And finally, at the bottom, we have the fork head box, a regulator of metastasis, and one of the key drivers in cell transitions from epithelial to mesenchymal. And this also shows clear patterning in colorectal and lung, with prostate being a bit more difficult to interpret. These are just some examples of what we can do with the power of RNA to capture these changes within just a handful of really well-studied markers. We also did proteomics analysis on these same FFPE sections, so serial sections, using mass spectrometry. A single 10-micron curl was prepared for proteomics analysis, and those resulting peptides were collected using both DDA and DIA mode. In the next few slides, I'm going to highlight these data, but at the left is a principal component analysis where the data are colored by tumor percentage, where 0 is normal adjacent, and then the higher ones are within the tumors that we were querying. And this is an excellent example to take you back to section 1, where we think about metadata consideration, because we do start to see differentiation by tumor percentage as we start to have lower and lower percent tumor. We see those samples creep back towards our negative group of 0%. So something that we always have to keep in mind when we look at these types of trials and cohorts. So we performed a t-test on the FFPE proteomics data comparing stage 1 to their normal adjacent tissues. And that's what this volcano plot is displaying, proteins that are up in stage 1 or down in stage 1. And when we consider these pathways, most significant changes are indicative of tumor progression, protein trafficking, stress response, ribosomal biogenesis, and metabolic adaptation, which is well known and something that we expected to see. So one of the things that we wanted to do is align our proteomics to RNA-seq data. And that's always in our field historically a bit challenging, partially because they are often discordant. And that's often because these are dynamic systems. They're snapshots in time from two deeply interconnected processes. Was it being translated? Was it already translated? Is the transcript down? Like all of these things are so deeply within the dogma that a lot of the times this is what we expect. And so what we're actually showing here is not true integration. It's each omic analyzed separately for concordant trends. So as I said, we really rarely expect the exact transcript tracked in both proteomics and transcriptomics in a way that allows for like an apples to apple true comparison. But so we can't do things like build correlation plots or similars. But what we do have the ability to do is to study those pathways that are affected and general trends, which is what this table and this string map is showing. So take, for example, proliferation. Although the exact transcripts and proteins aren't monitored in both omic, the pathway is strongly in alignment. And this is true for many other of these mechanistic properties, which enhances our understanding of what's happening on the system, even if it's not the exact same transcript to protein. And as a note, our ability to concordantly use omics in this way requires that each omic brings sensitivity and depth to an analysis. And so in this next slide, I'm going to talk just about LCMS data. So as technology and sample preparation and our analytical instrumentation advances, we're really able to provide a comprehensive proteome analysis. And historically, if we look in the literature 10 years ago when we talked about these types of analyses, we considered discordance between transcriptomics and proteomics. Proteomics was often maybe blamed for a little bit of that variability. And so I'm presenting this slide to show you how far this field has come with both sample preparation and, more importantly, instrumentation, which is allowing for us to do deep peptide level identification. In this cohort, we identified a median of 9,000 protein groups from FFPE blocks with a handful of outliers, and greater than 6,500 protein groups from plasma with mass spectrometry alone. These are incredible data, and I just want to reiterate to the folks here today that proteomics is really capable of providing deep and consistent analyses. So if that was bias that you had coming into multi-omics, I assure you that with our instrumentation, it is not there today. And that's quite important, I think, to just directly say. Our plasma proteomics is also very deepened. It's just such deep, beautiful data now. And we can deepen our findings by adding more omics to our proteomics analysis. I talked about this earlier, but this is the entirety of this expanded cohort analyzed by both Olink Explore HT and the Seer Proteograph with Orbitrap Astral Mass Spectrometry. And this is still just considering all cancer versus normal, but we can make many more observations than just cancer versus normal. This is stage 1 versus stage 4 in the plasma, where we've integrated those two omics data together. And we received one thing to maybe note here is that our stage 4 patients were post-treatment, right? They're folks who are very sick. And many of the people who are in stage 4, they've received a plethora of different treatments because they're very sick individuals. And there are several key signatures that come out when we start to look at these data in terms of the stages of disease. And here on the right, we're highlighting some of the key pathways that came out of it, where stage 1 really features proliferation and immune response, early the body's response to cancer. And in stage 4, we're looking at things like immunological evasion and adaptation, as well as adaptation to that therapeutic stress. And it really is another beautiful example of how we can see therapeutic potential from within these data. I can display these data a bit of a different way for you, also with a string map showing that the pathways identified by both mass spectrometry and Olink. It's kind of the same story as transcriptomics to proteomics, where we may not necessarily measure the exact same protein, but within some of those, we're hitting the same pathways. So this is colored where purple is mass spectrometry, orange is Olink, and then gray is both. So you can see that although it might not be apples to apples, we're really coming to the same conclusion. In the context of this talk, we previously discussed how important it was to bridge biomarkers in our discovery to non-invasive sampling techniques. And the reason of that is for patient care, comfort, and then compliance. And one thing that we are often overlapped is, all right, so what? What's the overlap? And in this analysis, we had interrogated over 11,000 plasma proteins, and that was by combining Olink and the Seer Astral Mass Spectrometry. And we also had analyzed about 12,000 protein groups from the FFPE. These are two very deep proteomics data sets. And they represent a total of 15,867 protein groups that we measured from this cohort. And about 48% of those groups have the potential to be monitored non-invasively. So to stress this Venn diagram again, within the same patient, we queried their plasma and we queried their FFPE from the tumor. And we had 48% of the proteins that were measured in both. And that gives us therapeutic potential to measure these biomarkers non-invasively, even if our discovery was from FFPE. It's incredibly exciting data for us, and we're looking forward to mining these much, much more. And that was it for the entire cohort. This is a great slide. This is it on a per patient level. Here we're considering each match set individually, and the Venn diagrams scale with their overlap. So you have the normal adjacent sometimes in the plasma, and then the tissue with the cancer tissue on the top. Normal adjacents on the top, cancer tissues on the bottom. And they're split out by disease area, including lung cancer, prostate cancer, and colorectal cancer. This is incredibly deciding data because it's really leading us into personalized medicine. And there's so much that we can do. There's so much more that we want to do in this type of study, and more that we could show you, but we don't have time. First, this is an incredibly exciting concept whenever we're able to translate direct sampling to indirect or non-invasive biomarker detection. It's huge for us as a field for what we might be able to do. And second, we're still working on integrating the bioinformatics using available multi-omics integration frameworks. And so that will come to where we're streamlining our LCMS data independent acquisition data into those custom FASTA files from the variants. The DIA data is our deepest. That's a data set where our Astral can deliver considerable performance. And, we want to utilize that, and we want to bank on that to really get us the deepest data that we can integrate. And finally, we're always conscious of how precious these materials are, especially in these studies with known outcomes that have longitudinal patient follow-up data. We recognize how precious a sample is where you've stored it for 20 years in order to get the follow-up data or the outcomes. So we're working to decrease our input material needs for deep analyses to ensure that we don't sacrifice data or coverage. We deliver deep data, but we minimize what we need as much as we are possible. So I want to, with that, reiterate some of our key learning points of today. We discussed the phenomenon of garbage in, garbage out for study design and how important designing a cohort that supports our hypotheses are, especially when we want to consider the resources and time that are required to collect these multi-omics data sets. And we really need to start with high quality samples that represent the disease. Next, we discuss a minimally invasive biomarker, why they're important for patient compliance, comfort, and cost, and then their use in multi-omics studies. And finally, we touched on the discussion of omics integration for concordance findings, giving support to analyses by parallel omics as well as true omics integration, where we require things like multi-omics integration frameworks. We use PAS, the Proteograph Analysis Suite for DDA data, something like Diablo for DIA. And before we move forward to questions, I really want to highlight why this matters. It matters because we believe that the future of healthcare is truly in personalized medicine. It's understanding on a patient to patient level disease. And that's so that we can understand how to intervene, so that we know what chemo to give, so that we can enable our clinician colleagues to make informed treatment plans. And in order to have the information to do that, we must also have the depth of knowledge about this one patient. And that's going to that need is going to lead us right back here to multi-omics analyses. And so it's truly critically important because this is the information that's going to drive better patient experience and ultimately better health outcomes for those people. And I think that for a majority of us here today, that's what we want to do and what we've committed our careers to trying to do. And so with that, I really thank you for your time and your interest in this topic. And I'm going to hand things back over to Corey. And we're happy to field any questions and continue this discussion.
Excellent. Thank you so much, Jessica. Really great presentation. And as you mentioned, we are now ready to begin the Q&A portion of the event. And as we do so, I invite the audience to continue sending questions or comments. Just a reminder, if you're attending via GoToWebinar, please use the questions chat box. And for those of you watching on LinkedIn Live events, feel free to post your questions using the comments tab that's there on LinkedIn. We have already received a few questions, and we'll try to get through as many as we can with the time that we have left. But rest assured, there will be an opportunity for follow-up after the webinar as well if we don't get a chance to address some of your questions. So this first question that we received here, Jessica, this audience member was asking about how much material did you need from the FFPE blocks?
Yeah, that's a great question. So one of the things we want to do is always minimize that. For our proteomics experiment, we're using just a single 10 micron curl with a product from Proteomics called the Beatbox with a downstream IST kit, and that's a really great product. And then we do the same thing from our genomics side, where we do a dual DNA RNA extraction. So even though I think 3 to 5 curls go in, you're able to get DNA and RNA data out of that common sample. And so I recognize how important that is, especially when you have things like skin cancer where you have a really small block. We want to get the most data that we can out of the minimal amount of input material. It's very important.
Excellent. Thank you so much for your answer to that question. Another question that's come in here, which I think is an interesting one and probably is far-reaching. This audience member was asking about what diseases or therapeutic areas, which ones are most likely to benefit from this multi-omics approach?
Yeah, I think that I think somebody asked a question in the pre-session about rare diseases. And we're in such a wonderful time to be scientists, where you have all of these databases coming online where you can query like normal ranges. Olink did the human proteome, right? And then you have tissue banks, things like that. And so I think that we can find that as we start to integrate more omics that we generate in-house, and also that the databases that some of our colleagues have published, like the UK Biobank. I think that things that have been historically difficult to power, once you deeply interrogate, mayhaps will come online where we can find markers of like rare diseases. But then I also think whenever we look at more popular diseases or things with more incidence and prevalence, we might be able to look deeply, like more subtyping, because that's one of the things that you always hear, right? Like if you're just like talking to folks about like cancer, they were put on a therapeutic, the therapeutic didn't work, they were put on another one. Like we want to end that. We want to say, let's characterize this biopsy, let's figure out, what the best therapeutic approach is, so that humans, right, don't have to endure that we can better match from deeply interrogating data to therapeutic options.
Fantastic. You mentioned cancer there at the tail end of your answer, and we had a question come in with someone talking about how, considering that multi-omics approaches have been used in cancer biomarker discovery, an audience member was wondering, do you think it's possible that similar strategies could be applied to identify biomarkers for targeted drug delivery in infectious diseases, for example? So they mentioned malaria, possibly addressing both the parasite and the human host overexpressed biomarkers. Do you think there's an application there as well?
Oh yeah, absolutely. I have some colleagues who are critical care doctors, and they talk about how they need to make a decision in sepsis, right? Like, am I going to knock the immune system down, or am I going to augment the immune system? And they don't have time, right? Like, this is a little tricky for multi-omics, but for fast therapeutics to say, like, I have to make a second decision about whether to augment or knock an immune system down, like whether or not proteomics, transcriptomics can deliver that in the time frame. It's a kind of amazing, right? And that's some areas of clinicians, a need that they have that I think we can deliver upon. Your question is really amazing, and that's whether or not we can use multi-omics in a bit of a different way to say, can I query a pathogen proteome simultaneously with host? I love that. I think we can absolutely do it. I'm a proteomics person. I probably maybe should have led with that most of my experience is proteomics. And now that we have our current generation of mass spectrometers that deliver that speed and sensitivity, I absolutely think we can start to pack a pathogen and host in a single analysis. And I think that's a really exciting time if you're a host pathogen person to be able to get those data and interrogate them.
Awesome. Great answer to that one. We have another question here that's come in. Pretty simple question, just wondering about the study you've been talking about through the presentation. Will it be published?
Yeah, absolutely. That's always one of our goals.
And then another question here, asking more specifically about, were you able to identify any new biomarkers or targets not reported on in the past through all this?
Oh, I'm sure that they're in there. When you have big data, you really have to sit with it for a long while to look at pathways and things like that. So I'm sure the data is there. I'm sure that we're going to find all sorts of really cool things whenever we start to deeply interrogate these data. But I think that one of the, maybe we leave with this, right? One of the great challenges of the field when we look at like our string maps or our pathway analyses is that we're only interrogating based on the tools we have, right? So we're only able to map things to string based on canonical human, which is based on the genome, right? The proteome is much more dynamic, right? So we have things like post-translational modifications, we have these variants. And so inherently, when we make these types of graphs, we're compressing them back down to gene level information, though we have many, many more. And so that's where I think that the field is you'll see it come up, right? When we get more proteomics specific database tools, so that we can expand our data understanding based on peptide level data. For we certainly have that data, we just have to use the tools that are currently available.
Speaking of tools currently available, we had a question come in, and I think we have time to hit a few more questions here before we wrap up. But to our audience, if we don't have a chance to get to all your questions, there will be an opportunity for follow-up after the webinar. We'll, of course, pass all your questions over to the Discovery team, and there's an email address currently up there on screen as well, info@dls.com, that you can reach out to. But I think we've got time to hit a few more. And this question, interesting, and maybe you can't reveal all your secrets, but audience members asking, but how were you able to streamline your proteomics workflow? I mean, it is typically pretty labor intensive.
Most of this is kits, so we didn't do a whole lot of work there. Like the Beatbox is a kit, anybody can go buy it. And so a lot of that is our vendors, right? The support that we get from our vendors is incredible. So really, in terms of our innovation, we're not doing a whole lot in terms of sample prep, right? Like we're just leveraging these tools back together. to be able to show you these types of data. But there's really not a whole lot of special sauce. Like Olink Explore HT is a kit, the Beatbox is a kit. Those vendors have done the work to really deliver these products, and we're just using them.
So that actually is a perfect segue to another question we had, which was asking about, for instance, the Olink and the MS platforms. How do they complement each other, and why not just choose one and kind of streamline your process?
Yeah, that's an excellent question. And this is something that we're seeing finally get published in the literature. And so most folks, whenever they do an Olink assay and then mass spectrometry with something like SEER, something like the Proteomics Enrich, Dynabeads, right? Um, the overlap is like 10 to 20%. I think 15 is about average from what you're seeing published in the literature. And that's also true for our hands. And so you're querying distinct regions of the proteome. And I recognize that there's some investment to do two big omics. But whenever you see that overlap and you see that you're getting completely different proteins, I think it's worth it, especially if you're talking about something where you have a small study size and you need to interrogate as deeply as possible to find that biomarker. We found that it truly expands the depth of protein coverage when you do multiple proteomics approaches.
Okay, and I think that's going to bring us to our last question that we'll have time for today. And I think it's a great question to end on. Audience members asking about how scalable this workflow is for really larger cohorts or for clinical trials. Like, for instance, could this be applied to thousands of samples in an efficient way?
Yeah, I do. And this is crazy. Like even in my time in proteomics, how far we've come. If you look at like Olink HT uses 176 samples per kit. So it's designed for throughput. But really, from the start of my career in grad school to now, seeing the advancements in the mass spectrometry's capability. So the mass spectrometer